very effectively. And the optimum temperature for Botrytis infection is between 15 and 25 Celsius, which is probably similar to what we have here in Western Washington. Presence of free water, which is rainfall, or even high humidity can facilitate an infection. So it's not surprising that Botrytis fruit rot is the main problem in raspberry production here in Western Washington. I would also like to talk about already gained knowledge how Botrytis infects red raspberry. So what do we know about the infection process? The main hypothesis that we have right now, that was established about 50 years ago, that fungus infect raspberry flowers that you can see here through its female part carpel, and doesn't show any symptoms on flowers. So stays latent in the flowers until fruit starts developing and maturing from these flowers. And then on fruit, we can see pretty distinct symptoms of gray mold. And based on this hypothesis, the main management strategies here in Washington and all over the world are built on. So here is a quote from Pacific Northwest Plant Disease Management Handbook that suggests growers to start spraying first at 5% bloom as soon as flowers get open, since we think that fungus infects flowers and stays in flowers until fruit ripening. And repeat spraying every 7-10 days, depending on weather, especially pre-harvest and especially if the weather is wet, since we know that disease is very conducive by wet weather. That's what growers do here. So all the disease management strategies are built on a calendar basis, and they start spraying from the time when flowers open all the way through the harvest. However, this hasn't been, there's not much research data on testing different fungicide programs time-wise, depending when to start spraying in raspberry system. One of the research that we were able to find in the literature was from Ohio State University, where they actually compared 3 types of spray programs, including full seasons that you can see here. Another one was sprays were restricted only to bloom period of red raspberry. And the third one, the plants were sprayed only pre-harvest. As a result, in the result of their 2-year study, when they looked at the disease incidence, they could see that in the program where the sprays were focused only on bloom, the disease incidence of Botrytis gray mold was the highest, comparing to full season and pre-harvest that actually didn't really differ between each other. So this gave us some food to think about, that how important our flowers actually are for the Botrytis infection of red raspberries. And is there anything that can be or should be changed in the spray programs that currently applied? As I mentioned before, we started working on this problem from 2015, and here I just briefly going to show you previous findings from 3 years of our research. So in order to see at which developmental stage fungus infects plants during the raspberry season, we divided the whole process into 7 developmental stages, so starting from close green buds all the way to the ripe fruit. And 2015 and 2016 summers, on a weekly basis, we kept coming to this field and kept sampling all these stages that were present on the field and seeing in how many samples there was Botrytis. Surprisingly, what we saw, that the level of colonization of flowers was very low comparing to the colonization of fruit. And we see that with flower and fruit development and maturation, percentage of colonization of these samples was increasing. And this data was consistent over already 3 years. Since I mentioned that wet weather is important for the disease development, we also looked at the rainfall data. So let's focus first on this 2015, 16, and 17 years data. This is accumulated rain from the beginning of April till the middle of August of these 3 years. So we see that during flowering in 2015 and 2016, there was not as much rain as comparing to 2017, when the amount of accumulated rain was higher during flowering season. Again, if we think about main hypothesis that fungus is thought to infect flowers, so this rain must have facilitated in better infection of flowers. However, in our data, we didn't see the increase of flower infection even when it was more rainy than previous years. If we look at this black line, so this is rainfall data from 2012 year. So we haven't worked on Botrytis gray mold that year, but what we know from growers is that that was probably last bad Botrytis year here in Western Washington. And when we looked at the rainfall data, we can see that the amount of rain during the flowering wasn't very different from, for example, 2017, but it was much more rain during fruit development and harvest. That makes us think that this additional rainfall during harvest may actually induce infection of fruit directly. And seems to be pretty important since gray mold this year was pretty bad here in Western Washington. I'm gonna switch to the projects that we worked in 2017, 2018. Since we didn't see a lot of botrytis in flowers, we decided to test how fungicide sprays protect these flowers. So we tested the effect of standard fungicide program that is used by growers here on Botrytis colonization of red raspberry. And we also wanted to test, do floral infections lead to fruit infections in raspberry fields? So we compared Botrytis isolated colonized flowers to that colonized fruit using genetic markers, just to get to use a different technique in order to test the same hypothesis that we were working on before, using more precise markers. This is the experimental design of the projects that we did in 2017 and 2018. So we picked two fields on commercial farm here in Whatcom County, and each field had plot which was left non-sprayed with fungicides at all, and the second plot was sprayed with fungicides as a regular program as all other fields get sprayed. The only difference between these fields was the age of plants. So field number 1 was younger compared to field number 2. In this experiment, we decided to focus only on 3 stages of raspberry flowers in fruit, not all 7. So we picked fertilized flowers or late flowers, so flowers that start becoming fruit, green fruit, and ripe fruit. So we went to these 4 plots over 2 summers and sampled these 3 stages and checked for how much botrytis is in these stages. If we look at the effect of fungicide applications on botrytis colonization, again, on the x-axis, our 3 stages, on the y-axis, percentage of samples that was colonized for each of the stage. What we see, first of all, about the same amount botrytis colonization of these late flowers comparing to previous years. But also what is important, we didn't see any difference in colonization of the flowers comparing sprayed versus non-sprayed plot. When we looked at the colonization of green fruit and ripe fruit, it was again consistently higher comparing to flowers, and they were statistically significantly different, sprayed versus non-sprayed. But again, if you look at the sprayed plot, about 80% of green fruit and 90% of ripe fruit had botrytis in them, even on sprayed plots. Again, and this data does not support the hypothesis that flowers are most important route for the fungus to cause disease on raspberry plants. If we look at the colonization of flower parts by Botrytis, so we dissected flowers into this female part carpel, sepal, and stamen, and looked in which organ there was the most amount of botrytis among those flowers that had fungus inside them. And our data was consistent with previous findings in research and previous findings during 2015 and 16 years. So if there was fungus in flowers, it was mostly in female part carpel and much less in other parts. But also, it didn't differ between sprayed versus non-sprayed plots. If you look at colonization of ripe fruit by botrytis, and here I am showing data for two fields separately, and in a second I'm going to explain why. So we looked at different morphological part of fruit to see at what part we had the most amount of botrytis and if it differed between sprayed versus non-sprayed plot. So our druplet, which is fruit itself, which is in the most interest of growers because that's what we harvest and sell, while stamen, sepal are receptacle as well. So in field number 1, if we look at the percent of samples that were colonized at each of these morphological parts, we see that first, fruit itself, almost every fruit had botrytis in it. If we look at the field number 2, we can see a little bit different picture. So the non-sprayed plot, 100% of fruit had botrytis in it. But the sprayed plot, only 30% of fruit had botrytis in it. Why this difference can be so big between these two plots? We speculate that the reason is the age of the plants. Since the field number 1 was much younger, it had much more dense canopy on plants during those years. Comparing to the field that was older, it had less leaves and less vigorous growth. So maybe that affected the efficiency of fungicide coverage of plants. So that's something that it can be, again, it can be checked and studied more, but something that can be also taken into account, that if age of plants and age of fields, on young fields, there probably will be more disease and infection just because we cannot really protect flowers and fruits that are behind the leaves. If we look at this data in a little bit different way, so here we look at quantitative data, average number of botrytis colonies per ripe fruit. Because as I said before, when we looked at flowers, if flower was infected with the fungus, there was usually just one colony per flower. So only from one part of the flower, the fungus grew. But when we looked at the number of botrytis colonies per ripe fruit, again, field number 1 and field number 2 separately here for the same reason, we see that in unsprayed plot, in fruit itself, the average number of colonies, so places from which fungus grew from one fruit, was about 6 from field number 1, while on sprayed plot, it was a little bit more than 3 average. If you look at the field number 2, the difference even bigger. So the number of colonies, average number of colonies per ripe fruit in non-sprayed field was again around 6, while comparing to sprayed was about 2. So what it tells you, this data shows us that fungicide applications seem to be effective at the fruit stage, even though we didn't see that effect at flower stage. So they do protect fruit, maybe not from infection, but at least from fungus growing out of the fruit. So this data shows that sprays are effective on ripe fruit stage. So over this few years, what we saw low rates of colonization of raspberry flowers. Botrytis colonization increases as fruit develops. And fungicide sprays didn't affect Botrytis colonization of flowers. That's what we've seen over these 3 years. And this led us to our second project to work on, to compare fungus in flowers and fruit using genetic markers. Because since it's a microscopic organism, you can't really take two individuals and put them next to each other and look at them and see are they different or no. But using molecular markers is much easier to do. For this, we asked the question, are fruit infections a result of flower infections? And we considered two scenarios. Scenario number one, that fungus infects only flowers. And scenario number two, that fungus infects multiple stages of red raspberry. So let's think about the scenario number one. If fungus infects only flowers, so let's imagine on a field we have some genetic diversity of the fungus, and each of this arrow represents a different genetic type, colors of the arrow. So for example, flowers get infected by two genetic types, black and red. What we expect to see in fruit if consider this scenario? Well, we expect to see the same genetic types in fruit if we think that all the infection came from flowers. Scenario number two, what if fungus infects multiple stages? So we have some genetic diversity, some genetic types that infect flowers. Then later in the season, we have the same genetic types that infect flowers, but also some new genetic types. Here is, for example, green arrows that infect fruit come later to the field. And on a ripe fruit stage, we have even more diversity. More genetic types came to the field and infected our fruit. When we looked at our data, what we saw? First, on the x-axis, 3 stages that we looked at. And on the y-axis, average number per inflorescence of colonies of Botrytis and pathogen diversity, the genetic types. Because for this project, what we did, we picked 20 inflorescences, marked 20 inflorescences on each of 2 fields. And we first sampled 1 flower from this inflorescence. Then later in the season, we came back and we sampled ripe fruit from this inflorescence. And later we sampled green fruit and ripe fruit from this inflorescence. Again, asking a question, if all the infection comes from flowers, then all of these genetic types should be similar, should be the same in inflorescences. But with this data, we didn't find the evidence of such case, because the number of colonies, again, increased with fruit development, and also pathogen diversity, so number of genetic types increased with fruit development and maturation, and was the highest at ripe fruit stage. What would we expect to see if scenario number 1, when only flowers get infected, would be the case? Probably that the genetic types that we found, again, in flowers would be the same that we found in fruit, or at least there would be overlap of the genetic types. But when we looked at this data, we saw that, so this yellow circle represents the, like a pool of genetic types that we found in flowers. And they didn't overlap with either green or ripe fruit genetic types. This area between green and ripe fruit that overlaps a little bit, it shows the amount of genetic types that was found in green and ripe fruit. So about 1% of the genetic types was shared between green and ripe fruit stage. But majority of genetic types in ripe fruit was different from the other stages. Again, it makes us think that fruit may be getting infected additionally later in the season, but not through flowers. So in the conclusion of this work, what we found, Fungicides applications do not decrease Botrytis colonization of flowers, either by incidence or quantitatively, but decrease amount of Botrytis cinerea in ripe fruit, so they're effective on ripe fruit. Diversity of Botrytis increases as fruit matures. Different types colonized flowers, green fruit, and ripe fruit in the same inflorescence, and that suggests independent infection events throughout the season. If we come back to our fungicide spray programs, what it means. So if sprays do not protect flowers and are not affected on flowers, maybe we could reduce amount of sprays that are applied on raspberry fields. And what it would mean for the industry? Well, first, it would save money because you don't need to spend as much money for fungicides. But one of the most important problems, it would decrease fungicide resistance. A very big problem that growers have right now, that because we spray so much, these fungicides lose their efficiency because pathogen gets resistant to it. So if we decrease amount of sprays, it will be the best strategy to cope, to manage the fungicide resistance. Of course, for this, we would need to do more studies and to do more field trials, because we also know that these last few years were pretty dry and not very conducive for the disease. But as for now, with this climate, that's the picture that we've got. I also wanted briefly to introduce another project that kind of this data led us to start working it. Since every raspberry fruit is infected by at least one colony of Botrytis, but we don't know the quantity, and we don't know when it starts affecting quality. So based on this information, we decided to start a project to quantification of Botrytis in processed raspberry fruit, since majority of fruit that is grown here is grown for processing. And that's a project we started working this year. So the current method that is used in processing facilities is hovered mold counting. When you smash raspberry fruit and put it under a microscope and try to find mycelia hyphae in order to quantify how many of them are there. There are a few limitations of this method, however. First of all, you really need to know what you're looking at, and you need to be able to recognize is it a mycelial hyphae or of the fungus, or is it the hair of the fruit. And also, you can't really distinguish between mycelium of other fungi, not just Botrytis. So it's not as precise. Because of that, we propose to start working with this project using Botrytis-specific medium that we use in previous projects, and also quantification of Botrytis amount in macerated raspberry fruit using molecular method qPCR. So we don't have any data on this project, but we just started working on it. And hopefully next year we will be able to share results with you. So that's the project design that we are going to use. We are using field samples that go to processing plant, and the same samples will be used for hover mold, for culture non-selective medium, and a qPCR. And then we'll be able to compare the data from all these three projects together. There are a few more projects that we are going to start working next year, since we got Washington Specialty Crop Block Grant to work on novel diseases, novel disease control strategies for berry growers here in Washington. And these projects will be focused more on monitoring and management of fungicide resistance, since, as I said before, this is a very big problem, and we need to develop high throughput methods that will help to detect fungicide resistance, their types, and how we